Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Yali Dou from the University of Southern California on this show. Please let me briefly introduce you to our audience. You got your PhD from the University of Rochester in 2001. You then moved on to do a postdoc with Robert Röder at the Rockefeller University. You then moved to the University of Michigan as an assistant professor in the Department of Pathology and Biochemistry in 2006, where you became associate professor in 2017 and full professor in 2020. In the same year, you then moved to the other coast, to the University of Southern California, where you are Marion and Harry Kuiper Chair of Cancer Research, Associate Director for Basic Science at the Norris Comprehensive Cancer Center, and Co-Director at the Brown Center for Cancer Drug Discovery. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
I think it's kind of strange for me. When I was very young, I always want to be a scientist. So when I grew up, everybody who's smart are becoming mathematician or physicist. And then, but by the time I get to the high school, all the smart people want to do biology. I think it has something to do with really the development of all this technology and all the molecular biology, all the tools available. So all of a sudden, everyone wants to become molecular biologist. So that's at the time that I'm considering college application. And then that's my choice.
Okay. So let's come to your science. And very broadly, it centers around MLL, KM2, and histone methyltransferases and their involvement in cancer and developmental syndromes. You started working on MLL1 during your time in the lab of Robert Röder. And there you started to characterize its function. For example, you looked at the interaction of MLL1 and MOF. Can you talk about what you found there?
Yeah, so I think when I started my postdoc career, I decided to do something with the chromatin and its function in transcription. So at the beginning, I tried to work on polycom because at the time, polycom was just discovered to be a histone methyltransferase. But then Bob Rader told me that it's so hard to study transcription if you study a repressor, right? Because there's so many things can go wrong. It's hard to figure it out for a negative experiment. So then at the time, I just have the first publication out that Jay Hess and Bevalis published a paper in Molecular Cell talking about MLL as a K4 methyltransferase that's implicated in transcription activation. So I was like, wow, that's great. So I can study something that's due transcription activation and still has something to do with histone modification. So that's how I got started working with the MLL, and turns out to be a great project as we still have a lot of unanswered questions. So back then, because it's Bob Rader's lab, that everybody start from biochemical purification. So my job is to purify the ML complex. So I learned how to do conventional chromatography and tried many different fractionation, tried to get the complex. In the end, I got successful by tagging the protein WDR5, which is conserved in mammalian and yeast SET1 complex. So from that purification, we actually purified, I still remember we got 22 proteins that's from that purification, and MOF is one of them. So then I decided, because MOF is the enzyme that doing dosage compensation, which actually I was also very interested in when I start looking for postdoc position. So that immediately caught my attention. So I set up the in vitro transcription assay, try to study the coordinate function between ML K4 methylation and MOF-mediated H3K4 acetylation. So, and then that's my first publication on MLL. I started this career-long pursuit of the function of MLL from there.
Yeah. So then the next obvious thing to do is when you purify factors is to put them together again. And this is exactly what you did. So how many factors did you actually need of those 22 to reconstitute the MLL core complex? And what did you learn from putting those complexes or those components together again?
Yeah. So at the time, I remember vividly that Bob Kinston Lab just published a paper, reconstitute the PRC1 complex. So I actually really got inspiration from that effort. So I was like, we know ML set domain has very weak activity. So how can we reconstitute the whole thing? If you look at the 22 protein, every one of them actually being followed up by later studies. So, but then when I look at those 22 proteins and see how conserved they are with the E-SET1 complex, there are not many, right? There are only 4 proteins or 5 proteins that's actually conserved between the human mammalian complex, E-SET1 complex. So that makes my job a lot easier. So I choose the first, I think 3 of them, the highly conserved ones. So I start doing the molecular expression and then put them together and then turn out to have great activity. And later, I think other people also try to reconstitute the complex using the bacteria expressed protein. And that makes things even easier. So that actually lays the foundation for all the later biochemical characterization and also the structural characterization of the complex.
I will get to that in a minute. It seems that during your postdoctoral work, you spent a lot of time in the cold room then, right?
Yes, a lot of time in the cold room.
Then you started your own lab and continued working on MLL1 and MOF. The first paper that then came out was about the crosstalk of H3K4 and H3K79 methylation that is regulated by another histone, namely H2B K34 ubiquitinylation. So what got you into this and how does this whole regulation work?
So that's actually, so when I first started my own lab, I still try to do the reconstitution and then try to look for the functional interaction between ML and the MOF, right? So I have a great tool and know how to reconstitute complex activity. So that's how I started with the MOF complex, because the MOF complex that interact with ML WDR5 is very different from the dosage compensation complex. So I got interested in what's the difference between this non-canonical MOF complex and then canonical MOF complex. Then how can if I can reconstitute this non-canonical MOF complex and then try to see if it has any activity that's different from the canonical one. So long story short, so that got us into a lot of interesting ideas. One of them is like the non-canonical complex does not have the protein MSL2, so which has been suspected to has E3 ubiquitin ligase activity. So that's how we got started, and we realized that activity is not on H2B K120. So we started to look for the new activity of that E3 ligase. So, and then, so everything has to do, start with the biochemical characterization. I'm really proud of that work.
Yeah, can you go into more details about that?
Yeah, so I think after we find the activity is different, and that's tied back to our work with ML, because we know the H2BK120 can stimulate ML activity. So what happens to H2BK34? Does it really stimulate ML activity as well? And turns out the answer is yes. And also another interesting finding we have is that the H2BK120 or K34 dependent activation of ML only happens to ML or some other ML family enzyme, not to all of them. So that's the first time people show that different MLs can respond differently to the histone crosstalk. So I think that's the major finding of that work. That is, the ML3, for example, cannot be stimulated by H2B-K120 or K34 ubiquitination. On the other hand, ML1 can. So that actually lead to the question that what differentiate different ML proteins? That they seems to be similar, right? They all methylate H3K4, and yet they are regulated differently by the H2B ubiquitination.
So how are they different? I mean, are they different, or are they present in the same cell, or are they just expressed in different tissues, or how are they all different, those MLL complexes?
That's the most fascinating thing. All of the MLL are ubiquitously expressed. They are in all the cells. They're pretty abundant. There's 3 subsets of ML family, and to each pair, they have exactly the same set of the interacting proteins. So on paper, you would think that they have functionally redundant. And yet, if you delete any single one of the ML gene, it's embryonic lethal. So, so I think that's a very interesting question that still remains to be answered. What's differentiate them?
You then investigated the therapeutic potential of MLL1 in MLL with in MLL of MM401. So this is a small molecule or an inhibitor. Can you talk about this inhibitor, how you found it, and what it can do finally?
Yeah. So let's go back to my biochemical reconstitution work. When at the end of close to the end of my postdoc training. So as I said, I first reconstitute the ML complexes that we only require 4 proteins together. And then from very early on, I know the protein WDR5 is very special because when we mutate the WDR5 binding pocket with the ML, or at the time, it's known that this binding pocket for his H3 tail, the whole complex fall apart. So I know that pocket somehow that's involved in ML interaction, and also it's a key to really for the assembly of the whole protein. So at the time, I was on the job market and kept thinking about what can we do utilizing this information that this interaction is key for the complex to get together. And all of a sudden, I think there's a eureka moment is that we can actually target this pocket to develop something that's inhibit the ML activity. At the time, I didn't know that WDR5 ML interaction is just uniquely required for the ML1 activity, not for other MLs. At that time, I thought this could be something that's a universal inhibitor that's block K4 methylation. That would be really cool as well. So by the time I get to University of Michigan, my first job, I immediately start looking for collaborations that can help me to develop inhibitor that block this protein-protein interaction. So when I asked around, I said, who should I talk to if I want to get this done? And everyone basically said, if you want to get this done, the best person you need to talk to is Xiaomeng Wang. He's a medicinal chemist at the University of Michigan. So we actually started our over a decade collaboration targeting the ML WDR5 interaction. And we published the first inhibitor, I think, in 2014. I think that's the most specific inhibitor that targeting this protein-protein interaction. So during this process, then we realized that this interaction is uniquely required for ML1. Because when we do the reconstitution for other ML complexes, ML2, 3, 4, and Sec1a, Sec1b, none of them actually absolutely require WDR5 to get to be fully active. So, and then we realized we got something very specifically that specifically inhibit the ML1 activity. That give us a great tool actually to study the function of ML1 in different biological context.
And how is this connected then to the leukemia? Because those mutation probably will then cause some overactivity of the MML then, right?
Yeah, so the MLL mutation in leukemia is there are actually multiple MLL aberrations in leukemia. MLL can be rearranged. So one allele of MLL can lose the C-terminal part of the protein and then put on and get to the fusion take on the C-terminal domain of many fusion partners. So there are also MLL amplification in leukemia, and also tandem duplication of leukemia. In those cases, the MLL actually keep the C-terminal set domain. So our approach to leukemia is that we believe that because in MLL rearrangement, almost all rearrangement occurs in only one of the MLL allele. So which means the other allele remains intact. So our hypothesis is that there got to be a pressure to keep that wild type allele so that they support the cell proliferation with MLL rearrangement. So that's indeed the case because I think Xianxin Hua's lab at University of Pennsylvania, they published a mouse study. So when they delete the MLL wild type allele in the context of MLL rearrangement, the cell proliferation goes down significantly. So around that time, we said, well, great. So that's really give us application to our MLL inhibitor. So we tried 7-8 different MLL rearranged leukemia cell lines, and all of them respond greatly to the MLL1 inhibitor. So suggesting that this inhibitor could function through targeting the wild type allele to achieve the leukemia inhibition. So although the later study by Patricia Ernst shows that the MLL deletion in MLL rearranged leukemia may not completely abolish or inhibit the leukemogenesis. So you need MLL2 also in that process. So I think it's possible that targeting MLL1 only inhibit some of the leukemia proliferation. We need additional compound, but no matter which cell line we tried, all of the MLL rearranged leukemia actually responds greatly to our inhibitor.
Did you move on with this inhibitor? Did you follow up on that work?
Yeah, so we are actually moving on. A lot of other people start publishing the WDR5 inhibitor. And also the recent study shows that WDR5 using exactly the same pocket can interact with many other proteins as well. So I think there's blocking WDR5 may not only block MLL1 activity. It could block other WDR5 interactive protein. So I think that may not give us the cleanest compound. There could have a lot of other on-target activity that has nothing to do with MLL1. So now we're taking a different approach to target MLL1 activity specifically that's independent from this WDR5 inhibitor.
And what approach is that?
So that's ongoing work. I'm not going to disclose yet.
Okay. You then further looked at the effect of MLL1 and MLL1 fusion proteins on leukemia transcription programs. So what do those proteins do to the transcriptional programs?
Yeah. So I think because MLL1 and MLL rearranged leukemia, they all keep the N-terminal, 1/3 of the N-terminal protein, right? So the N-terminal part of MLL fusion and N-terminal part of wild-type MLL, they have the ability to interact with DNA. So that leads to this long-term hypothesis that the MLL fusion and the wild-type allele all targeting the same genes. For example, the Hox genes, they all target Hox genes. That leads, for some reason, the MLL fusion bring in additional activities such as DOT1L, such as ELL, ENL, elongation, super elongation complexes that further enhance the activity. So you need this additional boost for HOXA9 expression, and that is really important for leukemogenesis. So that's the hypothesis. But when we look at the ML binding site with ML fusion binding sites that's published by Scott Armstrong's law, we actually see only 1/3 of them localized to the same genes. So 2/3 of the ML wild type targets are very different from ML fusion. So that has not been really mentioned or discussed in the field. So the ML wild type and ML fusion could do very different things. So what contribute to their localization at target gene is still open question.
Okay. So one thing that is tightly connected to transcriptional programs are enhancers. And an upstream factor of MLL proteins that promotes also leukogenesis is a pioneer factor, and this is HOXA9. You also worked on that, and I would be interested in what is the function of HOXA9 and how is it connected to MLL proteins?
Yeah, so it's quite interesting. The HOXA9 has been considered in the leukemia field for quite a long time. So 50% of AML has HOXA9 upregulation, and yet we know very little about HOXA9 function. So we did the work. First of all, try to look at where HOXA9 binds. It binds mostly to distal enhancers, and then there's 2 types of HOXA9 binding enhancers. One of them is one group of them is actually a common between the normal hematopoietic cells and leukemia cells. They all bind to those. But then there's another class of HOXA9 enhancer in leukemia. So that depends on HOXA9 to establish this leukemia-specific enhancer. Those enhancers are not actually activated or K27 isolation marked in normal hematopoietic cells. So we consider those as a leukemia-specific or cancer de novo enhancers that's dependent on HOXA9. And if you look at those enhancers, right, so they are mostly embryonic loci that's once bound by HOXA9, but then shut down in most of the normal cells, differentiated cells, or adult cells. But then in leukemia, they somehow get reactivated by this elevated expression level of HOXA9. So then we try to follow up on those genes, like how HOXA9 is able to establish those cancer de novo enhancers. And turn out that HOXA9 can interact with ML3 and 4 at those distal regions. And if you knock down HOXA9, a lot of those distal enhancers start losing the H3K4 monomethylation and K27 acetylation. So I think that's how HOXA9 really contribute to the transcription program in 50% of the leukemia cells.
So this leukogenetic influence of HOXA9 is really a concentration-dependent thing then? So the more HOXA9 you have, the more those embryonic enhancers get activated because of the pioneering activity of HOXA9?
That certainly contribute to it. So I think HOXA9 binding, that's one aspect of it. But when we look at the cancer-specific HOXA9 binding sites, those regions are quite dynamic, much more dynamic than the physiological enhancers, where you have HOXA9 binding regardless of whether it's, in the normal hematopoietic cells. So all these cancer enhancers, somehow, they really rely on HOXA9. If you don't have HOXA9, it shuts down. But in the normal hematopoietic enhancers, if you remove HOXA9, it doesn't matter. I think that those enhancers could be maintained by other hematopoietic transcription factors. So I think that's why I think the elevated HOXA9 certainly help. And also the local chromatin configuration could also help. So what contribute to those dynamic enhancers? I don't know.
Okay. So let's leave it at that. A next thing I want to get into is, and I think you already touched upon this in the beginning, because you were part of a team that did cryo-EM on the human MML1 core complex bound to the nucleosome. Was that what you were referring to in the beginning? That you are doing other things to get to the MLL function? And maybe can you talk about this project a little bit? How you got to the structure and how, what information you got from that structure?
So we actually publish a lot of structures along the way on individual ML components. And as I said, so the reconstitution really gave us a great starting point to understand how the ML activity is regulated. So we did a lot of work, but in the end, I felt like if we don't have a structure, we will never understand how ML is regulated, because I just cannot imagine how it works without the structure. So we really try to put a lot of effort to characterize the ML whole complex, and most recently the ML complex on the nucleus core particles. So this is really a very productive collaboration between my lab and the lab of Ansu Cho, who is actually learning how to do the cryo-EM work himself. So it's very, it's very courageous of him to start the first cryo-EM project in his lab by working on the ML core complex with the nucleosome. So at the time, we know that the H2L and the protein DPI30 contribute significantly to ML activity on the NCP. So, but we just cannot figure out how does it work. So by the time that he actually first saw the cryo-EM structure, he told me that how ML could position itself above the nucleosome dyad. And I felt like, wow, that's really makes a lot of sense, because the ML is actually very dynamic on the nucleosome core. It's a lot of rotational dynamics that's very different from other chromatin modifying enzymes. And DPI30 and H2L really helps to clamp down the ML complex on the NCP so that it won't be as dynamic. So that makes it access to the actually tail much easier for both tail to access to the active site much easier. So that really explains why the ML on the nucleosome particle, the activity of ML on the nucleosome particle is regulated by DPI30 and H2L. So the structure makes a lot of sense to us.
Yeah, I mean, you just went over the Nature Communication paper that was a follow-up paper to that by one sentence. So the functional implications on how this dynamic nature of the MLL and nucleosome core particles are unclear, and you got into that. Can you maybe talk about this in a bit more detail? How you worked on that and what the results were?
Yeah. So this is really as I said, so we did a lot about chemical calculation of the complex. So we basically try test every fragments of all of the components and see what's going on. From the very beginning, we know the H2L is very special among all ML complex components, even more so than WDR5, because the H2L has a lot of intrinsic disorder region. That protein, if you do the serious deletion, it really show like different. So basically, its regulation on the its regulation of ML activity on nucleosomes is so drastic that I cannot imagine how this intrinsic disorder region really contribute to the activity of the ML complex. So at the time, I have a great postdoc fellow, Yang-Tae Lee, who is actually an MR structural biologist. So he actually did a lot of work with H2L in the solution. So he actually showed that when the H2L get incorporated, and then also with the nucleosome core particles, those dysregulated region all of a sudden become structured. So that gave us the clue that maybe the IDR region, upon interaction with ML, upon interaction with the nucleosome core, will adopt a new structure so that allow it to engage in interaction that was not available if nucleosome core is not there. So that's actually got us. That's why I said I cannot imagine how it works without a structure. Mm-hmm. So really give us incentive to go after the ML structure on the NCP. And then later, once we have that structure, now we can visualize how this IDR region work, right, in the context of the NCP. Because upon those interactions, this IDR form a secondary structure that engage in the interface of the nucleosome particle. So that's contribute to stabilize the whole ML complex and try to limit its rotational dynamics of the complex on the NCP.
Is this dynamics in any way energy dependent, or is it all physics?
So I think that ML is very different from other chromatin modified enzymes. Other enzymes, very rarely you see all this kind of dynamics. So the question is really, is it functional relevant, right? So that's what we want to know. But if you look at the distribution of K4 methylation, it's also very interesting, right? You have regions that can have only monomethylation. You have regions have trimethylation, dimethylation. And yet, if you test all of the ML family enzymes, they can all do mono-di-trimethylation. Even though kinetic could be a little bit different, but they can all do mono-di-trimethylation, which means if the enzyme bind to a locus long enough, you should all see trimethylation. So if that's the case, why you have monomethylation-rich regions, right? Even ML4, they can do trimethylation in vitro and also in vivo too. So then why you have all these regions, enhancer regions, have all the monomethylation? So I think there are great localized regulation of ML activity, and this rotational dynamics really make it possible, right? If you think about it, if you push the binding configuration away from the nucleosome dyad configuration, then only one tail have access to the active site. So for a processive enzyme like ML, that means the capturing of the tail is actually harder. So that could influence the higher methylation more. So I think that's how you get localized the control. And another thing is the IDR of H2L, if you think about it, it only adopt this secondary structure upon interaction with DPI30, upon interaction with some other parts of the ML complex and also NCP. So how about if you get rid of DPI30, right? Then you don't have the secondary structure of the H2L. That means the ML complex will become very wobbly on the nucleosome. So that will limit the the trimethylation of the complex. So if you think about that way, then DPI30 mainly bind to the promoter. That's why you see more of a trimethylation. DPI30 bind less at the enhancer. That's why you see more of a monomethylation. So that's one way to explain it. But is DPI30 the only protein that does this job? That's the question.
So you already said that you don't want to talk about ongoing work. So if you don't want to answer my next question, that's totally fine. So imagine you are due to submitting a grant proposal tomorrow. What would you have written into that?
So we actually just got a grant funded that's based on our most recent publication at Nature Cell Biology. That's the first calculation of the non-canonical substrate for ML. So that's we are now actually not only just interested in the K4 methylation by ML. Actually, we're looking for additional function of ML in methylating non-canonical substrate. So I think that's really a open field. So our study on ML methylation of borrelin is really the first discovery of the non-histone substrates. I believe there are more non-histone substrate to ML1. So I think that's the direction and then my lab is going to take for the next couple of years to explore how this non-canonical function really contribute to the role of ML in cancer, in stem cells, and in other important biological context.
Okay. So for the last 35 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
Oh, no, it's really a pleasure to talk to you.
Oh, thank you very much. So then I think that's a good point to end this interview. Thank you, Yali, for your time and for being on the show.
Thank you so much.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.